So I spent the last 30 years focused mostly on Lymphangioleiomyomatosis. And in the process, we get referred patients with diseases that can look like it, especially chylous and cystic lung diseases. So we've seen a number of these diseases you've heard about today. This is the first time anybody said LAM at this meeting. So I won't go through the list of various diseases, but I always call either Denise or Adrian to run patients by, because I never see enough of any of these other things to be confident in my management. But it's been great to be in Cincinnati, where we have a really strong lymphatic group. And just on the right, just to remind everybody that about 3.5 liters of chylous fluid comes from the abdominal cavity, and perhaps about 0.5 liters from the lung. And chylous effusions occur when there's obstruction at the level of the thoracic duct above the pulmonary communication with the thoracic duct, pulmonary thoracic, and pulmonary lymphatic communications with the thoracic duct, causing reflux of chyle back into the lung, or loss of valve sufficiency, or direct communications through anomalous fistulas or vessels with the axial lymphatics. So I was going to start with a little review of LAM, because I'm going to go full circle through PAM and back to LAM. This is a low-grade, hormonally responsive, smooth muscle neoplasm that causes infiltration and cystic destruction of the lymphatics in the lung, leading to chylous effusions, recurrent pneumothorax, and chronic respiratory failure. In the upper right, you see this CT showing this tremendous profusion of cysts throughout the lung. Just below that, a chylous effusion, and on the lower left is a photomicrograph of LAM lung showing this infiltration of smooth muscle cells forming nodules and lining these cystic structures with tremendous remodeling of the lung. Early on, it was noted that LAM recurrence occurs in the transplanted lung, and suggesting that this was a metastatic neoplasm. That was first described in the 90s. And then just the last couple of years, there was a single-cell RNA-seq experiment done in Cincinnati that indicated that the source is most likely the uterus. And this was samples of explanted LAM lung at transplant, where so a single cell was performed on 3 different lungs and 1 uterus. And there was a population of cells, I don't know if the cursor shows here, but there's a population of about 100 cells out of 100,000 total cells that had a typical LAM signature expressing all the usual LAM markers. And in addition, when we asked the question, what other organ does this transcriptomic profile look like, it had a tremendous similarity to the uterine transcriptome. Of course, everyone knows that LAM is caused by mutations in tuberous sclerosis genes, either TSC1 or TSC2, but usually TSC2. And these proteins, or these genes, express proteins that regulate mTOR signaling. The LAM lesion expresses many lymphatic markers. In addition to the smooth muscle marker SMA, a LAM lesion expresses LYVE1, ploidoplanin, PROX1, VEGFR3, CD31, and a melanocytic pathway marker called PML100. The antibody for that is HB45, and that's a very useful diagnostic biomarker for LAM. The LAM lesion also expresses VEGF-C and VEGF-D, and you can see that in panel B that these LAM lesions get carved into an archipelago of these small LAM clusters that are lined by VEGFR3 expressing endothelial cells. And you can actually find these clusters of endothelial cells in lymphatic fluid and chylous fluid that are surrounded by a single layer of lymphatic endothelial cells. So one hypothesis for what destroys the lung is that a lesion develops somewhere in the body that expresses VEGF-C and VEGF-D. These cells move to the lung, express lymphatic or recruit lymphatic endothelial cells, cause this frustrated lymphangiogenic remodeling of the lung, forming cysts. We found that VEGF-D levels were elevated in women with LAM, and really none of the other cystic lung diseases that mimic it. And those include emphysema, pulmonary Langerhans cell histiocytosis, Birt-Hogg-Dubé, Sjogren's follicular bronchiolitis, and healthy controls. So this is a very useful diagnostic biomarker for LAM with a sensitivity of about 70% and a specificity of about 98%. And along the bottom, you see some of the cystic lung diseases that can be confused with LAM. So the steps in LAM pathogenesis, this is still a hypothesis, but I think a very solid one, is that a lesion develops somewhere, most likely the uterus, that has cells that are have mutations in TSC2 that cause proliferation, and then this lymphatic cleft formation that provides these clusters with access to the lymphatic tree, which then find their way to the thoracic duct, and the conducting lymphatics embed into the thoracic duct wall, proliferate, bud, and do that in a serial way, leapfrogging all the way up the thoracic duct. And we find this at autopsy. And these these LAM cell clusters reach the level of the neck and the anastomosis between the thoracic duct and the subclavian vein, and enter the venous system, and are distributed to the lung microvasculature, where these clusters wedge and invade at their leisure, which is one of the reasons we think that LAM targets really only the lung and the lymphatics. Is that there's this filtering mechanism, the lung, that allows these very low level neoplastic cells to invade. So we years ago, we did a trial, the MILES trial, which demonstrated that sirolimus stabilizes lung function in LAM. You can see in the panel at the right, patients who took the drug sirolimus had no change in their lung function over the course of a year. Those that took the placebo lost 10% of their lung function. And in the second year, when all the study articles were held, the sirolimus group declined at the same rate as the placebo group. So this drug is suppressive. It does interrupt lung function decline, but to benefit from it continuously, you have to take it continuously. It improves some measures of quality of life and some symptoms. It decreased serum VEGF-D levels. You can see that in a normal VEGF-D level is about 350. The placebo group median was around 2250 and stayed level throughout the course of the trial. The VEGF-D level dropped in the patients on sirolimus and stayed depressed, although there was a little bit of an uptick in the observation year when patients were off drug. That's more apparent on the spaghetti plots from this chart. And a subsequent study done by Joel Moss demonstrated that sirolimus is quite effective for the treatment of chylous effusions. That was published a month after the MILES trial. And the MIDAS registry that we've been undertaken since 2017 and been following patients on this therapy has shown very good stabilization of lung function. The average rate of lung function decline is actually 0, compared to a historical rate of decline of about 90 cc's per year in FEV1. So we're very confident that when we do finally get this data fully analyzed, that it's going to show durable stabilization of lung function. But recurrent pneumothorax remains a source of morbidity. Talc pleurodesis is the most effective method for reducing recurrence, but it's also the most fraught because patients develop chronic thoracic pain. They suffer future bleeding at future lung transplant, and talc is contaminated with lead and sometimes with asbestos. So it's really, we don't like using it. We'd like to have a better alternative. And now I'm going to take a little bit of a U-turn and talk about a completely different disease and bring it back to LAM. I actually saw a patient with this disease, pulmonary alveolar microlithiasis, in the adult clinic before this infant arrived for lung transplant, but I'd already been interested in it. We'd started to develop mouse models for it before this child was referred for lung transplant to Cincinnati Children's Hospital. And you can see this tremendous hyperdense infiltrate on chest X-ray and CT, this incredible profusion of these concretions within alveolar spaces that at lower power have this lamellar appearance as if they're incorporating alveolar components. And we've done proteomics and lipidomics, and they in fact have proteins in them that are derived from the alveolar space, and also some proteins that are found in renal stones. Won't have time to talk about that. But when we did single-cell RNA-seq on this lung, we found that the macrophages had a tremendous osteoclast signature. And I'll go over these genes in a little more detail on the next slide, but just have to trust me that these are signature genes for osteoclasts, and they're very upregulated in this infant's lung. So osteoclasts are derived from circulating monocytes, and I won't go through every one of these different organ systems, but if they are recruited to the lung and CG and CSF, they become alveolar macrophages. If they're recruited to bone and they see rank ligand, they become osteoclasts. And the same paradigm applies for intestinal macrophages and Kupffer cells in the liver and microglia in the brain. The binding of rank ligand to the rank receptor sets off a program of differentiation that includes multinucleation and polykaryon formation, and finally results in the expression of multiple genes that are important for degrading bone. So here you can see that usually osteoblasts are the source of rank ligand. They secrete this protein that binds to rank on the surface of the circulating or recruited monocyte. That sets off a gene program that includes expression of integrins, which mediate binding to bone, a cytoskeleton, a reorganization called actin ring formation that forms a little suction cup, such that the cell binds to the bone and then secretes acid and matrix degrading enzymes into the space between the bone and the cell, called Halship's lacuna. And I have to apologize for this next little video because I usually don't have sound, so I don't have to worry about the sound that came with this when I clipped it from this pharmaceutical website. But I thought it was kind of cool to watch these osteoclasts move across the surface of the bone, degrading the mineral component and the matrix underneath. And I hope this isn't too loud.
Osteoclasts play an important role in liberating minerals and other molecules stored within the bone matrix. Bone tissue.
Now, in order to be able to move, the osteoclast secretes a protein called TRAP, tartrate-resistant acid phosphatase, and it removes phosphate from osteopontin and other ligands on the surface of the bone so that the osteoclast can detach and keep moving. So TRAP is another very important marker for osteoclasts. When we looked in this infant's lung, there are many multinucleated giant cells expressing TRAP and the calcitonin receptor, which is a specific marker for osteoclasts. That were enveloping stones within the lung. There were also some mononuclear cells that were expressing these markers as well. So we made a mouse model by knocking out the transporter that mediates the efflux of phosphate from the alveolar space when surfactants catabolized, and they develop a beautiful mimic of the human disease with these hyperdense infiltrates and air bronchograms running through them. And when you isolate these microliths from the lung, they're perfectly spherical. They look like little cantaloupes, and they have a surface that looks like bone. And in fact, they are made of hydroxyapatite, and they are bone. And when we look at the gene signature and the macrophages isolated from these mice, they have a upregulation of all the usual suspects for osteoclast markers. And on immunohistochemistry, in the central, in column beginning, in this column, you can see multinucleated giant cells in the mice. It's very touchy. That stain for TRAP and cathepsin K, as well as the calcitonin receptor. And then again, in the PAM patient, the same markers are present there. And if we take these microliths out of a knockout mouse, isolate them and put them down the airway of a wild type mouse, they upregulate osteoclast genes, just as the same genes that I've showed you on the prior slide. If you look at the immunohistochemistry on the opposite panel, on day 0, these particles spread around the lung. By day 7, they've been gathered into these macrophage islands. By day 28, they're all gone. And that's, we believe, is due to this osteoclast action. In CCR2 null mice that can't recruit monocytes to the lung because they become trapped in the bone marrow, the clearance is much less efficient. So we think you need recruited monocytes to make this happen. So the overall hypothesis then in the lung was that if a circulating monocyte is recruited to the lung with MCSF and then sees GM-CSF, it becomes a pulmonary macrophage. And if it sees rank ligand, it becomes an osteoclast and secretes acid and matrix degrading enzymes important for particle degeneration. We wondered if the same thing happened in the pleural space. And if so, would this be a better agent for pleurodesis than talc? Because if it indeed induces pleural fusion through fibrosis, the hope would be that it would be gone within a few weeks or months. So the hypothesis was intrapleural hydroxyapatite will induce pleurodesis that's comparable to talc, but will be cleared from the thorax by osteoclast activity. And the experimental protocol was that we took synthetically, commercially available synthetic hydroxyapatite particles, and put them down the airway of mice or talc, and then sacrifice them at various times. And then we do this semi-medieval procedure in which in a post-mortem mouse, we impale their chest multiple times to induce a pneumothorax to see if we've induced pleurodesis. And you can see on the right, next to this inverted mouse, the pneumothorax is the very black space, which is colored in purple to make it clearer. So the first point was, do these particles upregulate osteoclast genes, and indeed they do. Rank and rank ligand are the two key factors required for osteoclast differentiation. OPG is a rank ligand trap that regulates osteoclast formation, and MCSF and its receptor also shown here, but they're upregulated by hydroxyapatite and talc. And just for completeness, we also looked at silica in the lung and asbestos, and it seems to be a class effect that these particles upregulate an osteoclast program, which makes some sense. That you're trying to degrade something, you're going to throw acid at it and try to get rid of it. And in addition to these differentiation cytokines, such as rank ligand, multiple osteoclast markers are also upregulated in these intrapleural lavage cells, these monocytes in the pleural space. We see multinucleated giant cells in the pleural lavage fluid that stain with all the usual osteoclast markers. And these slides we'll go through quickly because it shows similar degrees of efficacy for both hydroxyapatite and talc, but after installation into the pleural space, both talc and hydroxyapatite increase pleural thickness. You can see this Masson's trichrome showing that there's an increase in pleural thickness on day 56, both for hydroxyapatite and talc, and it's virtually overlapping in terms of degree over time. And we can also count adhesions by opening the chest cavity and counting the number of strands between the lung and the chest wall. And actually, hydroxyapatite performed a little better than talc for the formation of adhesions. And in fact, hydroxyapatite induced pleurodesis just as effectively as talc using this transthoracic needle puncture technique I mentioned. Here, the colorized areas are the airspace, that's residual airspace after pleurodesis. And you can see that on day 7 and 28, there's a reduction in both hydroxyapatite and talc mice in the volume of the residual airspace. That's quite durable in the hydroxyapatite treated mice, maybe a little less durable in the talc treated mice. And it's completely cleared. This hydroxyapatite is completely cleared from the thoracic cavity by 2 months. You can see here by day 56, there's no residual radiodense particles, or virtually none left in the hydroxyapatite treated mice, whereas the talc is never cleared. We asked the question of whether osteoclast activity is important for the fibrogenic and pleurodesis effects of hydroxyapatite, and indeed in mice that have key osteoclast factors knocked out, such as the ATP6 knockout mouse and the CTSK knockout mouse, there is a reduction in the adhesion score on day 14. We didn't see any reduction in the adhesion score for talc, and that the mechanism of fibrotic fusion for talc may be different, more aggressive and different than for hydroxyapatite. Finally, we had a malignant effusion model in which we put Lewis lung carcinoma cells into the lung, and then 4 days later, we instilled hydroxyapatite or talc, and both treatments reduced the volume of residual pleural effusion on day 25 compared to controls. So getting back to your Dr. Seuss reference, so we started with LAM. We were very lucky to find that sirolimus helped these patients stabilize lung function, but pneumothorax still occurs. And then studying another very rare disease, came up with the thought that perhaps these hydroxyapatite particles in PAM might be a better agent for pleurodesis than talc. And indeed, these particles are rapidly cleared, and they seem to have equal efficacy to talc. So in summary, osteoclasts have not previously been known to play a role in lung homeostasis or pathology. Talc and hydroxyapatite particles induce osteoclast differentiation in pleural monocytes. Osteoclast products, acid and matrix degrading enzymes play an important role in hydroxyapatite-induced pleural fibrosis, and hydroxyapatite particles are as effective as talc for pleurodesis, but unlike talc, are cleared within 2 months. So next steps are to complete the tox experiments in mice. We're going to demonstrate efficacy and safety of this treatment in pigs, and then approach the FDA to obtain an IND to do a first-in-man trial for refractory pleural effusions, a larger pivotal study, hopefully with company support for pneumothorax and effusions, and then someday, I hope, a trial in LAM patients. Most of this work was done by two very talented postdocs from Sapporo, Yusuke Tanaka and Yasuaki Uehara, both of whom have gone back to Sapporo, and now Yuki Takahashi has taken the reins and has started to complete some of these experiments. The single-cell RNA-seq analysis was done for LAM by Jan Zhu in Cincinnati and by Jennifer Franks and Cole Trapnell at the University of Washington. So thank you very much.